Hello, everybody. My name's Steve Diggle. I'm a microbiologist at Georgia Institute of Technology. And in this video, I'm going to tell you some of the things that my lab is working on related to cystic fibrosis infection. So one of the things to note is that I moved to Atlanta from the UK back in April 2017. It's been a great experience to move to the States, and the lab's very excited to be here. And it's been also great to be able to interact really strongly with Children's Healthcare of Atlanta and CF Atlanta. And we've started and are continuing collaborations between Emory and Georgia Tech, and that's all very exciting. So I'd like to tell you a little bit about what our lab does, and we're really interested in how microbes interact with each other, and particularly during cystic fibrosis infection. So why do we want to study infection? I mean, it may seem obvious, but one of the main reasons is that infection, whilst also causing a lot of illness and deaths, is the healthcare costs are astronomical. So this is an old report by the CDC from 2009, but even in 2009, you can see that healthcare associated infection in the United States alone cost up to $45 billion per year. And just the benefits of preventing infection could save upwards of $31 billion a year. So huge costs. Chronic wound infections alone affect 1 to 2% of the population, resulting in around about 200,000 deaths a year. And again, huge costs to the healthcare system. And this problem with chronic wounds is only going to get worse because chronic wounds are a problem in patients with type 2 diabetes. There's an epidemic of type 2 diabetes. And so we can expect to see more of this. And also these kind of infections are really dangerous for the evolution of strains that are resistant to multiple antibiotics. So it's a real problem we need to get a handle on. Jim O'Neill, you can see at the bottom, is an English economist who was instructed by the UK government to do a report on antibiotic resistance and what the problems were going to be if we didn't get on top of that in the future. So the conclusions really from that report, the takeaway conclusions are that by 2050, if we don't do anything about antibiotic resistance, there'll be 10 million extra deaths per year around the world, purely due to bugs that we used to be able to treat. This is going to be a huge cost to the global economy. Some estimates say $100 trillion per year. And therefore, what we need are new antibiotics. But this is a problem because drug companies are not necessarily investing heavily in new antibiotics because it costs a lot of money to do that. And then the bacteria get resistant very quickly. But we do need them desperately, or we need other drugs and other ideas. But more generally, I think that we need to understand infection a little better before we can really start to design new drugs and compounds. So I'm going to talk a little bit about what we do in that area. So I think when we think about an infection, and in particular in cystic fibrosis, where we have a chronic infection of the lung over many years, we need to think about infections as ecosystems. And if we're going to study ecosystems successfully and understand them, we need to do multidisciplinary work. So infections are ecosystems because we have intraspecies interactions. So these are actions that are going on between members of the same species. We have interspecies interactions. So we know that the CF lung is made up of many different types of bacteria. Sorry, the microbiome is made up of many different types of bacteria. And all of these potentially interact. Bacteria might compete, they might cooperate. So we need to understand interactions on that level. They also signal to each other, and I'll talk about that very briefly in a minute. They form these biofilms, and these are kind of where you get kind of basically slimy cities of cells, where they'll grow as millions of cells attached maybe to a surface or free floating in, say, sputum, and they surround themselves with a sticky, sugary matrix. And also we need to understand disturbance events like when we treat with antibiotics. And I would say thinking about infections as ecosystems is actually very poorly understood. And to understand it, we need to think about multiple ways to tackle this. We need to understand how questions. How do things work in bacteria? So we know that bacteria have lots and lots of different things that they can do, like they have flagella where they can swim, they make biofilms, they signal to each other. But what are the mechanisms of that? How do we, so when we're thinking about how questions, we're thinking about how does it work? This is where we figure out the genes that are important for all the different things that bacteria do. We also do a lot of genomics as a group and as a microbiology field, where we can sequence lots of different strains from patients. This is becoming of obvious importance during the COVID pandemic, where we're being able to identify particularly dangerous variants. And it's the same with bacteria. So we need to do a lot of genomics on clinical samples directly from patients. And this will also help us demonstrate and identify transmission networks between patients. But we also need to ask why a certain bacterial behavior is there. Not just how do they work, why are they there? What survival benefit do they provide to bacteria in particular environments like the CF lung? And so these are evolutionary questions, and they're different to the molecular mechanism questions, but they're also highly complementary. So by working with different people in different fields, we can get much more of a handle on understanding bacterial behaviors in general. We also need new models as to how to grow the bacteria. And what I mean by that is we need new models that are more realistic towards infection. We need the bacteria to behave in a similar way in these models as they do in infection. And so there's a number of groups doing that, including Marvin Whiteley's lab at Georgia Tech, who's created a synthetic sputum medium, which we'll be using later in this talk. We could also work with engineers who can do very cool things now. So they could engineer ecological landscapes, for example, and we could grow the bacteria in there. They can make smart surfaces that bacteria can't bind to anymore. So understanding the interaction between bacteria and surfaces is important. And it becomes important if you can design a smart surface on a catheter, for example, that bacteria can't bind to and cause infection. But it's also very important, and this is something that is really great between Emory and Georgia Tech, is close links with clinicians. Because clinicians obviously are dealing with infections and patients directly, and they understand the real problems that we're dealing with. Sometimes scientists could be working in a lab with a strain of their bacteria that isn't particularly a relevant strain that's out there in patients. And so by working together, you can kind of more accurately try and solve the problem that you're dealing with. And of course, we've talked about this. We need new drug development, antibiotics, and other ideas. So I'll start with this quote. So it's perfectly possible to imagine a rather boring universe without sex, without hormones, and without nervous systems, peopled only by individual cells reproducing ad infinitum. And this universe, in fact, exists, and it's the one formed by a culture of bacteria. Now, this quote was said by a Nobel laureate, Francois Jacob, in 1973. And he's basically saying that bacteria are boring. One cell divides into two, they don't do much else, and that's that. Well, we now know that this is really not correct. They have incredibly complex social lives. And I'll just outline a couple of things that we're interested in related to that. So we know that bacterial cells individual cells will signal to each other. And they do this signaling to coordinate different behaviors. And this has become known as quorum sensing. And I'll describe that a little bit more in a minute. But why would you want to signal to each other if you're a bacterial cell? Well, one reason is that you might want to protect a niche with your friends. You might want to coordinate virulence factor production, toxin production, so you can coordinate attacks on hosts. You might want to combat host defenses by signaling to each other and coordinating your behaviors. You may want a population migrate across a surface as a whole group of cells. Or you may want to hunker down and form these slimy cities we know as biofilms. And on the right, you can see a sort of a picture of a surface down here, and the green and red are different strains of Pseudomonas aeruginosa, the bug I work on. And this kind of big lump here is what you would call a surface attached biofilm. It's formed of many millions of cells. So one of the things I mentioned was that bacteria do something called quorum sensing. And this is really signaling between cells. And it's called quorum sensing because the bacteria wait until there's enough of them around, so a quorum, before they actually do a particular activity or behavior. And very basically, when bacteria are at low cell densities in an environment, so here's not many cells here, and they make a signal that they release, which is the blue circle here. So when there's not many cells, there's not much signal in the environment. And what happens is that the bacteria will grow to a high cell density. So here you've got more cells, you have a lot more signaling. And the signal accumulates in the environment until the bacteria are able to sense a change, a critical change in this signal in the environment, where they then know, because it's a proxy for how many cells there are, they then know how many cells there are, and they then start behaving like a population. And they turn on quorum sensing controlled genes and behaviors all at the same time, all at once. So if you think about that in terms of making toxins, the cells will wait until they're at a certain amount, a certain density, and then all of a sudden, there's enough signal in the environment, they know how many there are, and they all make it en masse. So this is a good way to say overwhelm a host. So they really do have quite sophisticated behaviors. And we now know, over 30 plus years of research, that quorum sensing signals are very chemically diverse. So here's just some. You can see that they're structurally very different. They're all made by different species of bacteria in these cases. But we also know that quorum sensing regulates a whole different suite of behaviors in different species of bacteria. So for example, and this is really where it was probably safe to say, this is where quorum sensing was really first described, bioluminescence, so light production in squid. So you know how squid make light in the sea? Well, actually, it's a colonies of bacteria colonizing their light organs. And bioluminescence in this bacteria called Vibrio fischeri is actually controlled by signals. And so they don't make light until there's enough of them there. So then people realize that these molecules are made by lots of different bacteria, some of which cause disease. So this is the bacteria we work on, Pseudomonas aeruginosa. Some of you may have heard of it, some of you won't have. But it's a gram-negative, opportunistic pathogen, which means it doesn't cause disease in most healthy humans. But it can cause a wide spectrum of infections if your immunity is breached in some way, or your immune defenses are breached. Some strains are transmissible, so they're highly transmitted around. And really importantly for this talk, it's the key cystic fibrosis pathogen. It's likely the bacterium that causes the most issues in patients with cystic fibrosis. But it's also hospital acquired. It often causes ventilator-associated pneumonia. So we can be expecting a big rise in Pseudomonas secondary infections from people on ventilators due to COVID. And the problem is it's intrinsically antibiotic resistant, so it's hard to get rid of. The CDC, therefore, considers it as a serious threat. And so it's one of the organisms they're most worried about in the future moving forward. From our point of view, I've spent many years studying its quorum sensing system, which is very complex, involving lots and lots of genes. But we won't go into the detail of that because that's it's like 30 years of research and lots of complicated diagrams. But the important thing about Pseudomonas is it can switch its lifestyle. So it can cause acute infections, i.e. in burns. And it's a really serious problem if it gets into a burn wound with a very high fatality rate of maybe something like 70 to 80%. And it's very rapid, causes like septicemia and things. So, and it does this because it makes a bunch of cytotoxic products like tissue damaging exoenzymes, exotoxins. It even makes hydrogen cyanide. So it makes cyanide. And it controls most of this by quorum sensing, waiting till there's the amount of cells there, and then with the signals, and then making toxins en masse. But when it gets into an environment like the cystic fibrosis lung, it starts to behave itself a little bit more in terms of it doesn't make as many toxins. What it does is it starts to think, well, I want to stick around in here. And so I'm going to have to form something that stops me getting destroyed by the immune response. So it forms these biofilms where it encases itself in sugary, sticky matrices, and it produces more things. It tends not to produce as many toxins in the CF lung, but it will produce more things that make it stick together and become a problem to get rid of. So it's very good and very adaptable at switching lifestyles. This is just a diagram to show you how many different things it makes. You can see over here, exopolysaccharides. It makes 3 sugary coatings that can surround the cell. Alginate is the one where we pull out colonies and they're very sticky on a plate from a patient. And this is due to alginate, and it makes them very gloopy. We call them mucoid colonies, but it makes 2 others called PSL impel. It makes a bunch of stuff. It's not worth going into any of these individually, but this is just to show you that it makes a lot of different toxins and ways to protect itself. So cystic fibrosis and infection of the airways. So I'm sure a lot of you know a lot about cystic fibrosis. So it's a genetic disorder. 75% of people are diagnosed by age of 2. And more than half now of the CF population is age 18 or older, because we've got better at treating, or clinicians have got better at treating some of the infections. And the new modulator drugs are very exciting as well. But characteristically, there'd be a sticky buildup of mucus in the lungs, the pancreas, and other organs. And from a bacterial point of view, the mucus clogs the airways and traps bacteria, leading to infection. And ultimately, the outcome of this is increasing lung damage and eventually respiratory failure. And one of the things that we're worried about with Pseudomonas aeruginosa is that there's rise of multi-drug resistant aerugenosa in CF patients. And you can see this here, that like as patients get older, well, patients get colonized very quickly with aerugenosa, but there's now a rise in multi-drug resistant aerugenosa. That's a real concern. So what are the challenges that we have to treat Pseudomonas infections in the CF lung? Well, we know that chronic infections last a lifetime, generally. And this is ultimately because antibiotic treatment is going to fail. I mean, it works in terms of patients can get ill, they're given an intravenous course of antibiotics and they make an improvement, but ultimately the Pseudomonas will hang around and lung decline will continue. And some of the problems we have as Pseudomonas researchers is we have a lack of appropriate models to study CF infection accurately. The mouse pulmonary lung model is not particularly in any way similar to a CF infection. I think there's a pig model, but Pseudomonas doesn't colonize the pig CF model very well, so that's not an accurate model either. And there's some good in vitro models that have been designed, such as the synthetic sputum medium from Marvin Whiteley's lab, that the bug Pseudomonas behaves in a similar way in there as it does in the CF lung. So these new models are exciting, and hopefully is going to give us a lot more information about Pseudomonas growth in the CF lung in the future. But one of the things we're particularly interested in is that Pseudomonas evolves and diversifies during long-term CF lung infection. And it's this diversity that we feel is important because it's there, and the populations are so diverse that like one strain can be in there that's doing something, and then the one next door to it is doing something completely different. So I'll try and explain through that. So this is kind of the title of this is Pseudomonas aeruginosa populations in the CF lung are phenotypically and genotypically diverse. So what do I mean by that? So CF patients tend to get infected by a single strain of Pseudomonas aeruginosa. So there's lots of potential strains you could pick up from the environment, but in the CF lung, there tends to be one strain that gets in there and then starts to dominate. And as it dominates and it sort of expands, it starts diversifying. So all the phenotypes start to change. They start to look different. They start picking up mutations in their genomes. And so they become very diverse, even though they're from the same originating strain. So these 6 plates here just show you colonies taken from a single sputum sample from a single patient at one single time point. And you can see just by eyeballing them, each one of these colonies is Pseudomonas aeruginosa, but they look remarkably different. And if you sequence them, they're all the same strain. They've just diversified phenotypically and genotypically over time. So this creates as a problem because microbiologists and in both the research labs and clinical labs tend to only pick single colonies from sputum samples to work on or to test for antibiotic resistance. And this is a problem. So why does it matter? Because things like antibiotic susceptibility tests do not always predict resistance. So this is a graph just showing 44 colonies. So each dot is one of 44 colonies, or each data point is one of 44 colonies of Pseudomonas aeruginosa picked from a single sputum sample on one plate. So Sophie Darch, who did this in my lab back in 2015, just took a single plate, picked the 44 colonies off, and then asked the question, what happens if we do a susceptibility test to all these different antibiotics? Because remember, in the clinical labs, they may only pick one colony and do these tests. And then they give that information back to the clinician, who then has to decide the most appropriate treatment. So what you can see here, let's take ceftazidime here. You can see that the majority of these 44 colonies that were picked are actually sensitive to the antibiotic. So that's good. This red line here shows you the break-off, the cutoff point where colonies are considered to be resistant. And you can see that in this case, 3 of the 44 colonies were actually considered resistant using this standard test. So that tells you that if you picked one of those 3 colonies, and given the information back to the clinician, it would tell you that like the Pseudomonas strain was resistant to ceftazidime. But quite equally, you could have picked, or more likely, you would have picked ones that said that they were sensitive. So by picking only one colony, you are underestimating the sensitivity and resistance of antibiotic to antibiotics from Pseudomonas. So the question is what should we do? Should we take sweeps of plates, look at multiple colonies? I think this is an important thing to consider in the near future. Another thing that we're interested in is how does Pseudomonas aeruginosa form aggregates in cystic fibrosis sputum? So an aggregate you could think of as a type of biofilm. They're where cells group together, they encode themselves in a sticky exopolysaccharide matrix, and they're hard to treat with antibiotics, and they're difficult to eradicate. So what we find with aggregates, though, is that in sputum, they tend to be free floating. So they don't really attach to any surfaces. They free float in the sputum. And so understanding that is an important step into being able to eradicate them. So we grew 2 different isolates in a synthetic sputum medium that was designed in Marvin Whiteley's lab at Georgia Tech. And Pseudomonas behaves in a very similar way as it would do if it was grown in human sputum. So what we found is that with these 2 different isolates that we were looking at, is that they form 2 very distinct types of aggregate. We have ones where the cells sort of join end to end like this. We call this a stacked aggregate because they're just like looking like little ladders where they all join together. And the other one is what we call a clumped aggregate. And in this case, there's no organization. In the stack, they're quite highly organized. In this, they're not. They just all form giant clumps. They all just pile in. They form either giant clumps or very small clumps. So there's no organization. And the real interest to us was, well, why is this kind of so different between 2 different Pseudomonas isolates? So they're the questions. What is the mechanism of this? What does it actually mean ultimately for any treatment? Does it matter? So what we found with this is that both physics and biology are important for how Pseudomonas forms aggregates. And this is ongoing work. But it all involves, and like this is like a complicated looking diagram, but I only want to take a couple of points away from it. It involves polymers in the medium, and polymers are things like mucin and extracellular DNA. But it also involves the bacterial cell surface. So Pseudomonas surrounds itself, or on its surface, it has what we call lipopolysaccharide, or LPS. And the LPS can vary between isolates. But what tends to happen is many strains have what are called A bands and B bands that stick out of the cell surface. They stick out of the cell. And an A band is the same across all Pseudomonas aeruginosa strains. And it's just made up of rhamnose sugars, like joined end to end. They also make a B band, and the B band is variable. And it's made of different sugars and different combinations of sugars. And so the B band is what gives some of the different strains of Pseudomonas aeruginosa what we call their serotype. And they use serotype to diagnose certain Pseudomonas strains in the clinic. So what we really need to understand with this is that Pseudomonas can make an A band, or any cell can make an A band alone, a B band alone, an A and B band together, or no bands at all, depending on the genetics of the organism. So what we found with this is that the stacked aggregates actually have an intact B band. So they have an intact variably sugar B band. And the clumped strain we found had lost the ability to make a B band. So this was not expressed on the cell surface anymore. And we now have a working model for how all of this works. So if we look at the stacks, so overall, the aggregate phenotype is going to involve the polymer and what we call the hydrophobicity of the cell surface. So when we lose that B band, the surface becomes very hydrophobic. And so this kind of explains what we're seeing. So in the stacks, these cells have an intact B band, so they have a hydrophilic surface. And they stack because the polymers in the medium are pushing them together by a force called entropic deaggregation. And it's just pure physics. They get pushed together by polymers, like mucin. But in the mutants that don't make the B band, so they become very hydrophobic on their surface, hydrophobic cells will stick very closely to other hydrophobic cells. And they're able to overcome this entropic force by the polymers, and they then just clump randomly. Now, that's what we think's going on. We're trying to confirm this. But why is it important? Well, it's important because many, if not the majority of Pseudomonas strains taken out of the cystic fibrosis lung are actually LPS mutants. So they're likely to be ones that form these close little clumps. And so we really need to understand, are these more resistant to things like antibiotics than this type of aggregation, for example? So this is kind of new stuff, and we think it's interesting. The final thing I want to mention that we do is that Pseudomonas makes little things called Piosins, and they're like bacteriophage. So if you don't know what bacteriophage are, they're basically viruses that attack bacteria. And structurally, our Piosins look very similar to phage. And they're made by Pseudomonas aeruginosa to kill other strains of Pseudomonas aeruginosa. So they have a real potential in whether you can use them to treat Pseudomonas because they're quite potent. So each strain seems to compete with each other to try and kill members of the same species. So they look like little bacteriophage. Here's a bacteriophage here. Bacteriophage have a base plate. They have a tail, and they have they have these tail sort of these legs, tail fibers that will attach to a host cell surface. And then what happens is that the phage will come in, land on the surface, and then puncture a hole through the bacteria that it's attacking. And in the case of bacteriophage, they have a head which contains nucleic acid like DNA and RNA. Our Piosins are almost identical, except they don't contain this head. And so they're not able to replicate. So when Pseudomonas makes them, then it doesn't make any more than what it initially makes. Like they don't replicate like phage do, because phage is a virus, it wants to keep replicating. So what happens is that they're so potent that one Piosin unit can kill one cell. So they get made by a Pseudomonas strain, they find another Pseudomonas strain, and they land and attach to the surface. And what they then do is the tail fibers contract, the sheath comes down, and it punches a hole through the cell membrane of the cell, and it causes what we call membrane depolarization. There's a hole there, things leak out, the cell dies. And this kind of Piosin sheath has been described as one of the strongest molecular nanomachines known, with an incredible force of like 550 piconewtons during punches. So very, very potent. So they're really interesting biologically. The question is can you use them to treat infections? So I mentioned right at the start that we have a problem with new antibiotics being made. They're very expensive. Bacteria gain resistance to them very quickly. So things like phage and piosins are what we might call alternative therapies for infection. And phage have been used for sure with some success. I mean, and in treating actual people with infections, and they've been used successfully in a couple of cystic fibrosis patients. So both of these things have promise as alternative therapies. We can use them in doses. There tends to be no interactions with antibiotics. Both of them use the LPS as a receptor. So, we talked about LPS before, about making different aggregates. And a lot of CF strains don't actually have the B band that sticks out. Whether this actually matters for whether some of the phage and piosins can attach to those strains, we still have to investigate. So there could be problems using these in terms of some of the strains might be resistant. So phage, some of the problems with phage is they actually replicate. So, you could use them as a therapy. They'll kill the bacteria in there, but they'll continue to replicate because they have this nucleic acid head. And so they can continue to evolve, and they're hard to control. So is there a risk that they may go rogue and start attacking species of bacteria you don't want them to? So this is kind of one of the downsides. With our piosins, they don't replicate, so they don't evolve. They're highly specific to Pseudomonas aeruginosa. They're very useful for the type of piosin it makes to diagnose the strain of aeruginosa and which other piosins would be useful to kill that strain. They're very stable when you purify them. So they have real potential. It's just that not much work has been done on using them in actual patients. Okay, I'm going to finish there. So thank you very much for listening. This is a picture of my lab during lockdown. We were doing it appropriately with social distancing and things. If you're interested in any of the work we do, here's the website, and thank you.